Secondary growth in dicot stem. Secondary growth is defined as the increase in diameter or girth of the axis due to the activity of lateral meristem, which includes the vascular and the cork cambium. The cambium is a meristematic tissue which is present between the xylem and phloem of the vascular bundle. The vascular cambium consists of two different types, fascicular cambium and interfascicular cambium. The fascicular cambium is present between the xylem and phloem, whereas the interfascicular cambium connects two vascular bundles. So therefore, the cambium forms a ring-like structure. Since the cambium is meristematic in nature, it continuously divide and produce more number of cells. The cells that are produced towards the upper side forms the secondary phloem, whereas those produced towards the lower side or the inner side forms the secondary xylem. Also, the parenchyma cells which is present between the two vascular bundles becomes meristematic and they get elongated to form plate-like connections between the pith and the cortex. These connections are called as secondary medullary rays. Secondary xylem. It is also commonly known as the wood. It consists of two systems, namely the axial system and the radial system. In the axial system, the cells are oriented in the horizontal manner, whereas in the radial system, the cells are oriented in the vertical manner. The secondary xylem, as like the primary xylem, is also made up of tracheids, vessels, xylem parenchyma, fibers, and the xylem rays. The vessels and tracheids in the secondary xylem are shorter than the primary xylem and has calariform thickening with pits. In general, more secondary xylem is present in the stem because the secondary phloem eventually during the secondary growth gets crushed, and therefore only the wood forms a major part of the stem. Due to the continuous activity of cambial cells in the dicot stems, concentric layers of dark and light bands can be seen in the transverse section. These rings are called as annular or growth rings. The formation of these rings are influenced by seasonal changes. For example, the spring or early wood formed during the spring season is lighter in color because the vessels have a wider lumen during this formation, whereas in the autumn wood or late wood, the rings appear darker in color due to the presence of lumens that are shorter in the vessels. And the study of the annular rings and determining the age of the plant using these annular rings is called as dendrochronology. Heartwood and sapwood. In the vessels, the cross walls dissolve and form bladder-like ingrowths called tyloses. These tyloses block water conduction and hence the xylem elements in that region becomes non-functional, resulting in a dark central region in the wood, which is called as heartwood. Whereas in the sapwood, there is no blockage and therefore the wood is functional and lighter in color. The heart The secondary phloem, also known as bast. The formation of secondary phloem causes the primary phloem to crush towards the outer side. Therefore, the primary phloem becomes dysfunctional and later becomes bast fibers, which is a very economically beneficial forest product. And due to continuous production of secondary structures in older stems, the epidermis gets ruptured, resulting in the formation of a new protective layer called periderm. The periderm is made up of three tissues or the layers, namely the phellem or the cork layer, the phellogen or the cork cambium, and the phelloderm or the secondary cortex. First, the phellogen. It is a secondary lateral meristem that arises from living cells of the epidermis, cortex, hypodermis, or the phloem cells. It causes increase in diameter of the stem. The phellogen undergoes periclinal and anticlinal divisions to form the phelloderm or the cork layers. Whereas phellem or cork cells are compactly arranged and thin-walled initially, but later during the development, suberin gets deposited and they become thick and elongated. For example, Quercus, the phellem of Quercus is used as bottle cork. Phelloderm is a parenchymatous tissue found external to the secondary phloem and hence is called as secondary cortex. The cells are arranged in definite rows and may contain starch. Then comes the bark. Bark is of two kinds, the ring bark and the scaly bark. In the ring bark, the layers form a complete cylinder or ring, example vitis, whereas in scaly bark, the periderm is not continuous but occur in separate strips, example Quercus. Lenticels. In the periderm, we can see small portions of called lenticels. Woods define these small portions as loosely arranged cells which are present to increase the activity of the phylogen or the cork cells and possess numerous intercellular spaces. Their chief function is they form the chief aerating structures and they usually occur near the older stomata. Thank you.